artificial intelligence will completely transform our world. But what is AI? Why will it affect you? And how can you and your business survive and thrive through the AI revolution? Welcome to AI and You. Here is your host, author, speaker, and futurist, Peter Scott.
Hello and welcome to episode 321. We are back with another fascinating interview this week. What is AI doing to medicine? Some years ago, before ChatGPT, I was getting frustrated and annoyed. I was telling students that AI would one day cure cancer because that was a constant refrain from the futurists, and I believed it would happen. But I was complaining, when are we going to get that? We were promised a cure for cancer, but all we got was recommendations on what movie to watch next. And then AlphaFold came along. Google DeepMind's invention, named in the same pattern as their AlphaGo program that became the best Go player in the world, made this enormous impact on the problem of figuring out protein structures, which we'll get into in the episode. And I went, yeah, that's what I wanted to see us using AI for. Far freaking out. Well, things have gotten more complicated and nuanced since then, and some of my frustration returned. So here to help us sort it all out today is Professor Mohammed AlQuraishi of Columbia University's Department of Systems Biology and a member of their program for mathematical genomics. He heads a lab focused on biological perspectives at the molecular and systems levels. He has an MS in statistics and a PhD in genetics from Stanford University and was a departmental fellow at Harvard Medical School. As if that wasn't enough, before this storied academic career, he founded 2 startups in the mobile computing space. He is one of the top researchers in the world in computational modeling of protein structure prediction. We're going to talk about Mohammed's background in molecular biology, the big goals of that field, and his North Star of understanding biology well enough to simulate it. Why I haven't yet seen the kind of revolutionary impact I was expecting from AlphaFold, what it means for a protein to fold and why it's a big deal. And if you've never seen a picture of a protein structure, Google one up right now for context. And how AlphaFold and similar models work at figuring that out. So here we go. Mohammed AlQuraishi, welcome to AI and You. It's a privilege to have you here and get some answers to some questions that have been bugging me for a long time. Before we dig into the toughest of those, let's get at your background a little. You have an intriguing bio, which at one point says that you knew even in your early 20s that you were too old to begin studying physics. And that's an attention-grabbing statement. What led up to that and what followed that?
Well, I always perceived scientists, particularly physicists, as being somewhat, if not prodigies, maybe the wrong word, but people who typically have some innate intuition for these questions in kind of physical sciences. And even in high school and elementary school, whatever, there were certain people that just seemed to have a very, very strong intuition for some of these questions that I felt like I didn't have. On the other hand, I felt I had intuition for other things, like computer science, or even entrepreneurship for that matter, which other people seemed not to. So I think based on these observations that I made just growing up, at that point in my life anyway, I felt like kind of the trajectory that I was on meant I would certainly avoid, let's say, high energy physics as a career path.
And what was your background in molecular biology, and how did you proceed at that point?
So actually, my background, truthfully, was very much computer science. I started programming at a fairly young age, and I very much kind of saw myself as somebody who was kind of native in the software world, in the computer science world. So when I began my undergraduate studies, I was focused primarily on computer science. But what happened sort of halfway through, I felt like I could do more. It felt like, computer science is interesting, of course, but it felt like I wanted to sort of broaden my horizon, if you may, and sort of integrate and incorporate other scientific areas. So at that point, I actually did something of a sort of systematic survey of different fields that were sort of topical at the time. And biology really struck me as, this was not long after kind of the Human Genome Project had been sort of solved and all that stuff. So it was very much in the news. And it seemed to me like biology was very intriguing because it was sort of asking questions that had the flavor of a computer science question. It had to do with logic, with control, with organization, with information processing. with the sorts of things that one often thinks about in computer science, but that were taking place in a very, very different context, in the context of the cell. So it felt like if I were to go into biology, I would be able to kind of leverage all this intuition I had previously from computer science, but then apply it in a new domain that was very exciting. And that was very much at the frontier of science. I added essentially an undergraduate degree in biology to an existing computer science degree, and finished with 2 degrees in 2 different topics, but felt like was well situated now to kind of begin thinking and working at the intersection of those 2 fields.
A lot of physicists are motivated by hunting for a theory of everything, a grand unified field theory of some kind. What was it about biology that excited you as some kind of brass ring? Is there something out there that's like, that is your life's goal, that is the thing you're working towards, you can encapsulate that excitement around?
That's a great question. And I should say, I actually had very much that same reaction when I did this survey. I was always, I should say, taken by physics, and I thought physics often asks the deepest questions. What struck me about biology wasn't its sort of depth of conceptual content, but really more the potential for so much, right? It's sort of the substrate of life. It's kind of the basis of all of the adversity that we see on planet Earth. And so it felt like the kind of space of things we could do, if we understood biology to the same extent that we understand physics, is really unlimited. Now, to answer your question in terms of, what is the kind of the North Star, the thing that really sort of excites me, I think it's this notion of, on the one hand, understanding biology well enough that we could simulate it. So essentially distilling our understanding of biology into a kind of a computational artifact that we can run on the computer, and that would essentially allow us to emulate all the behavior that we see in the real world, in the biological real world. So that's on the one hand, you have to kind of understand the potential for that understanding. And then on the other hand, having built that sort of understanding, using it then to, essentially build new life. It's sort of the equivalent of having an operating system in the computer science, but for biology, right? Really being able to engineer biology at scale and just generate entirely new forms of diversity.
Wow. Is achieving that going to mean merging biology and physics?
Possibly. It's an interesting question. So there are different ways to interpret that question, right? But one interpretation of this is to say biology so far is made up of organic compounds, right? And maybe a few metals. But generally speaking, the chemical repertoire of biology is maybe somewhat limited. And what it means to sort of go beyond that and kind of begin to integrate physics is that you actually start having quote-unquote biological organisms that are much richer and sort of broader in the way in which they're grounded physically. And that, I think, is certainly a direction that I would imagine biology taking, like sort of new life taking, moving in the future. There's also a version of this question which has more to do with the methodology of understanding the system. So physics is very mathematically grounded. And the question here is, would the study of biology become more like the study of physics in the sense of rigor and sort of theory building that we have? I think that's true. But I also think, sort of interesting, we living at this kind of transition point, right, in the kind of the pre and post AI world. It's clear very much that I think that AI and again, kind of computational thinking is going to permeate much of what we do. So I think in addition to sort of looking to physics as an analogy or as an inspiration, I also think computer science and kind of computational primitives are also going to be playing a very critical role in how biology evolves in the future, particularly in the sense of how we understand biology and how we model biology.
Well, one place where biology gets close to physics is protein folding, and I want to talk about that. And I want to lead off with a question that's been bothering me for some years, and so I'm going to take the time to put this out in some detail. In the process, I will erect a monument to my ignorance, which I expect and want you to demolish. But it's one which I think is grounded in a belief that a great many people would have. So you'll be doing a service to a lot of people by explaining where I'm going wrong here. So following the news at a distance from outside the molecular biology community, AlphaFold grabbed the headlines for, as I read it, having taken a field where understanding the structure of a protein, the three-dimensional structure of it, and we can explain in some detail the nature of that after this question, where that's a difficult thing. There are about 200 million known proteins. and the structure for less than 200,000 of them had been worked out. And according to what I was reading, it took either $1 million or several years or a PhD thesis to do that through some kind of X-ray crystallography process. So expensive, but clearly worth it for some reason, because the shape of a protein means so much. Its shape shows up in things like the protein spikes on a coronavirus. And if you know the shape, then you can construct something that binds to that shape and renders the virus inert. I realize I'm almost certainly stacking up thing after thing that's of ignorance here, but I'm going to keep going. The nub of my question here is, after reading that AlphaFold had essentially predicted what all of the 200 million proteins structures were and put that in the public domain, my expectation of the outcome of taking a field where the knowledge is 0.1% of what's available and turning that up to 100% is going to be revolutionary. I expect that this is going to result in, at some point, in not that long a time, in headlines that trace radical developments back to that moment. And I can't say that I have seen that. And yet, clearly, understanding the structure of proteins was important enough and valuable enough that people would spend $1 million for each one. So by any measure, this should have dumped tens of trillions of dollars of value into a market. And I haven't seen what I would expect to be visible outcomes of that. And I really want to know why. This is your point to start taking apart all of my fallacies.
No, I think it's a sensible construction. I think these are good questions. There are different ways to answer this. So one thing to clarify first is this sort of question of breadth versus depth. For things that have commercial value, for things that have therapeutic and medical value, right, they'll almost certainly have to concern proteins that come from humans, or at least proteins that are in, say, pathogens or things that we want to target and so on. So even though it's absolutely true that we know, in fact, now of billions of proteins that exist on planet Earth, and for which AlphaFold has predicted the structures of most, that huge pool, from the perspective of medicine or therapy or whatever, or commercial value, is much smaller, right? We don't, in a way, at least immediately, care or can convert the prediction of the structure of proteins in some exotic thermophile that lives, in a volcano into commercial value, right? That it doesn't have inherently that commercial value, at least not immediately. So even though, when DeepMind predicted essentially initially, a few 100 million protein structures, which from the perspective of basic science is incredibly enabling and empowering, it still remains the case that most of those predictions don't necessarily translate into utility for drug discovery. So that's sort of one aspect. The other aspect is more what I would call, let's say, kind of the depth of the prediction, or so let me explain by way of example. Essentially, there are corner cases from the perspective of a method like AlphaFold. Corner cases meaning predictions that are not the kind of the normal thing you do on a day-to-day basis from the perspective of the algorithm, but that are highly valuable. So for instance, if you put a protein in close proximity to a drug, such that the drug binds that protein, that affects the binding of the drug to the protein, changes the protein, changes the structure. Another example, if you were to mutate a protein, you introduce a mutation in the sequence that could have pathogenic consequences. It could make that protein make you sick. That has an impact on the structure of the protein as well. And it's by impacting the structure that actually the phenotype arises. But those things that I'm calling corner cases, the kind of the predictions that are off the beaten track. So on the one hand, they are central to the questions that have to do with drug discovery, because it's precisely under those perturbed conditions that disease arises and that therapies arise. Right? You're modulating the normal behavior or the abnormal behavior of a protein. So it's very much these kind of side quests, the things that the protein does that doesn't normally do, that become critical. And it just so happens that these side quests are actually the hardest things for AlphaFold to predict. They still, in large part, remain outside its purview. So one way to kind of summarize is to say, on the one hand, AlphaFold can predict 1,000 more structures or 1 million more structures than we had of the vanilla type. But of its ability to predict structures other than the non-vanilla type, and these happen to be the ones that have most therapeutic relevance, is far weaker. Worth noting here, AlphaFold 2 was the one that won the Nobel Prize. A couple of years, 4 years after, AlphaFold 3 came out, which expanded its purview. It could tackle some of these problems. But it remains the case today that some of the most interesting types of predictions we want to make are ones that are not accessible yet to AlphaFold-like models, because they add these complications that make it harder to solve the problem. And so that's one reason why maybe what you would expect to be kind of this massive commercial value hasn't really materialized overnight. I would also just say it takes time. And I think there will actually be a lot of commercial value and human health value emerging from this exercise, but it takes time. Drug discovery is also hard.
Thank you. I feel smarter already. And having established the value of this field, let's take the time to just look at what protein folding means. We have some idea of, I gather, the sequence of a protein, but not its shape without this complicated process of either crystallography or AI prediction in some way. And the three-dimensional structure of a protein, the visualizations of those are the insanely complicated structures that look like the output of a pasta-making machine on LSD. So there's just the space of possibilities of those shapes. We're not talking about origami and like the difference between folding a crane or a frog. It is countless orders of magnitude more complicated than that. What does, and we've been using this word prediction, so there's an element there of uncertainty of the output. So what does it mean, first of all, to be able to predict or how to predict the structure of a protein? What are the rules that a protein follows for folding itself? How does it know how to fold itself up? And then what is it about the predictions that make them more or less reliable?
So in terms of how it knows how to fold itself or how the folding process happens, maybe just to clarify, actually stepping back for a moment. So the process, generally speaking, is you start with a piece of DNA. So that's made out of the 4 nucleic acid bases, the ATCGs. And then there's sort of a multiple processes that happen in the cell by which that piece of DNA essentially gets converted. So the piece of DNA is one-dimensional. It's ACTG, just their letters in sequence. That gets converted into a sort of a different chemical basis made out of 20 instead of 4 letters that are also in sequence. They're also linear, right? So at some point, kind of early in the life of a protein, which is just this linear, initially just this linear sequence of amino acids, of these letters, in the early life of a protein, it's just a string, effectively. And then what happens is that string folds, quote unquote, on itself, meaning it undergoes various motions until it kind of takes on a well-defined three-dimensional shape. And that's what we refer to by folding, is that it goes from that kind of linear, thread-like appearance into something which looks more like a 3D object with volume and surface and so on. Now, to the question of how does it know how to do that, it actually depends a little bit. So for some fraction of proteins, it's entirely driven essentially by physics, by thermodynamics, right? So the amino acids, those letters, have different chemical properties. They have different geometric properties. So for instance, you can imagine if you have 2 amino acids, one of which is positively charged and the other is negatively charged, they might attract each other, for instance, right? There are other physical forces that materialize this process. But essentially, just by virtue of the kind of the physics that are encoded in the chemical identity of each letter in the protein sequence, that process drives it towards taking on a three-dimensional shape, at least for some fraction of proteins. It used to be believed, or I don't ascribe belief to necessarily to kind of the state of science at any point in time, but I think maybe broadly speaking, people thought this was almost entirely true of all proteins, that there's this kind of self-driven process. I think it's fair to say now, particularly for higher eukaryotes, including humans, that a lot of this is actually also facilitated. So there are often other proteins like chaperones, which act as sort of little incubation machines in which a protein in the kind of the stringy, unfolded state enters, and then it emerges as a folded and well-structured protein. So it's not entirely clear, or it's not entirely true, that this process is sort of entirely driven by thermodynamics. There's probably some facilitation that happens for at least some fraction of proteins, and potentially a large fraction of proteins. But either way, whether it's pure physics or sort of biologically assisted physics, the end result is you end up with a three-dimensional structure.
Does that structure represent, in some sense, a minimum potential energy configuration?
Yes, almost certainly. Strictly speaking, they may be what are called kinetic traps. So it's possible that there are yet lower energies, but they are so challenging to access that the protein never gets to, essentially. And so let's say from a practical feasibility standpoint, it is certainly a low energy state, most likely the lowest energy state, but not necessarily, strictly speaking, always the case. And often what happens, and maybe this is a critical point, is when, for instance, a protein binds another protein, or say a protein binds a small molecule, a drug, that lowest energy shifts. It becomes something different. And because of that shift, that causes a change in the shape of the protein. And so one of the most critical frontiers that science faces now in the molecular biology world is this question of how do proteins change in shape in response to binding other things or changing in the environment or what have you.
And I ask this because the problem of finding a local or even a global minimum in a multidimensional space is a well-known and one in computer science, and certainly tackled a lot in AI, like gradient descent. So is that the sort of approach that computer science brings to solving this problem?
In part, yes. So you asked earlier, why is this hard? And it's hard precisely for the reason you articulated, is that there is a kind of large space of possibilities in which a string can take on a shape. And so typically you're doing some sort of, obviously in principle, some sort of energy minimization, some sort of gradient descent that takes you from this high energy state to the low energy state. In practice, though, you can't really do this very naively. People historically, for decades, try to essentially apply physics in a very literal sense. So you have some notion of a physical energy function. When one state is high, the other state is lower. And you just essentially try to pretend like you're going down this slope, this gradient slope, down the energy, the physical energy gradient slope to the native state, to the lowest energy state. But the problem is, I kind of refer to this notion of a kinetic trap. There are kind of mini traps, right? Is that there are mini low points, mini minima along the way that you can get stuck on. And so if you simply were to kind of naively just go down at every step, you'll just get stuck in this local minima that doesn't represent the true global minima. And you have to kind of get kicked out of it, out of these, small wells to get to the bigger and bigger wells. Mm-hmm. What AlphaFold ends up doing, to the extent that we understand it, is it essentially, there are multiple things it does. And fundamentally, I think that the key idea is it builds a very kind of smooth version of this energy landscape based on coevolution. And it's a specific concept. I'm happy to get into it in more detail if you'd like me to. But the basic idea is that across evolutionary time, you could look at a set of related proteins and infer from their statistical properties, and that's something I can unpack, something about which amino acids are interacting with which other amino acids, such that you have a very good hypothesis as to what the structure of the protein is. So that AlphaFold, when it tries to make the prediction, it starts off with this really good hypothesis that essentially kind of smoothes this landscape, so that when it does kind of the equivalent of gradient descent, it can do it essentially in like a single shot or in very few steps.
I had supposed that it worked by being trained on the 190,000 odd protein structures that were already known and using those somewhat similar to how AlphaGo was trained on successful games of Go, and saying, well, these are configurations that match. Now extrapolate and interpolate those to how these other sequences might fold up. Was any of that part of the equation?
It is part of the equation, for certain. But it's not as big a part of the equation as one might think. In conventional machine learning settings, what he described is 100% the case, right? You have some training data, you have inputs, outputs, you learn a map, and you call it a day. In the protein structure case, it's actually only a part of the puzzle. So you mentioned the 190,000 sequences or structures. In the grand scheme of things, actually, that's not a very big data set for machine learning. But just to further drive home the point, my lab, we did this experiment in this system called AlphaFold, which is sort of the equivalent of AlphaFold, where we actually shrunk down that data set from 190,000, essentially all the way down to about 3,000 structures. So really, really tiny data set. And essentially, you could show that even with 3,000 structures, you get about 90% of the performance. So you don't diminish all that much. The model doesn't diminish all that much. And it has to do with this thing I was referring to earlier with this kind of co-evolutionary pattern. So one way of taking this is that what AlphaFold does doesn't really correspond to going from a protein sequence to a protein structure. That actually remains a very hard problem and an unsolved problem. What it instead does, it takes our protein sequence, finds a bunch of other sequences, other proteins, whose sequence is very similar, so they're evolutionarily related to it, uses that sort of set of related sequences to derive this guess that I mentioned, this really good guess as to where the structure is, and then using that guess, solve the problem. So if that step weren't present, if it didn't have access to these guesses, to these really good guesses coming from these evolutionary related sequences, AlphaFold would not have worked. We would never have had what we had or we have today. So that's really quite central to the entire enterprise. It's quite different from, predicting the sounds of text, like speech recognition or image recognition or vision. This is fairly unique to this domain, owing to the fact that proteins have evolved.
That's the end of the first half of the interview. Pretty intense stuff here, so we'll come back to this next week. You can see here my modus operandi, if you will, of basically taking things I have the biggest questions about and finding someone who's an expert on that to make it clear. And it should be clear by now why these questions about protein folding are so important. In today's news ripped from the headlines about AI, researchers at Meta, in their Fundamental AI Research, or FAIR, division, presented version 2 of Brain2QWERTY, QWERTY being QWERTY for first part of a typewriter keyboard, their system that can translate brainwaves into text. The team also included contributors from the French National Centre for Scientific Research and other French institutions. We've had what amount to mind-reading machines for over a decade, but this is taking it to a new level. This version can decode complete words and sentences. To train the system, they used a magnetoencephalography, or MEG machine, which is how I'll refer to it in the future, to record the brain activity of 9 subjects typing sentences in English, totaling 90 hours or 22,000 examples. The MEG is a non-invasive device that records a brain's magnetic activity. Brain2QWERTY version 2 achieved a 39% word error rate, the percentage of words guessed wrong, whereas version 1 was 43%. What's interesting about this is the discovery that training across multiple subjects' brain activities improves performance even compared to training on a single individual. This system can be used for restoring natural communication for people with some version of locked-in syndrome. Imagine what this could have done for Stephen Hawking, who could communicate at only about 2 words per minute on average. This new technology holds the promise of communicating at 90 words per minute. Next week, we'll conclude the interview with Mohammed AlQuraishi, when we'll talk about how the advent of AlphaFold landed in the protein folding community, how AI is about to transform molecular biology and scientific experimentation, the possibility of simulating a cell, OpenFold, which is Mohammed's answer to the closed-source parts of AlphaFold, and his vision for the next 10 years of the field. That's next week on AI and You. Until then, remember, no matter how much computers learn how to do, it's how we come together as humans that matters.
That's all for this episode of AI and You. Please leave a rating and comment and share with your friends. Get the book Artificial Intelligence and You and see more videos and articles at aiandyou.net. That's aiandyou.net, where you can also send us your questions. Thank you for listening.